Okay, welcome back, class. This is the last part, the third part of our lecture series on the physiology of vision, and it is entitled Central Neurophysiology of Vision. So I'll now share my PowerPoint presentation. Okay, so we're at the third part, the Central Neurophysiology of Vision. Okay, let's now study the principal visual pathways of vision. So when the object gets registered in the retina here, it now goes out through the optic nerve. Okay, so the nasal fibers cross to the other side to end up in the visual cortex, while the temporal um, registry would temporal registry would stay as is on the ipsilateral side of the eye. Okay, so it's only the nasal registry which will cross. Okay, after okay, so these two optic nerves will now meet at the optic chiasma here, and there's crossing of the nasal fibers. Okay, and then the nerves coming from the optic chiasma will now go to your lateral geniculate body, and this is now your optic tract. Okay, optic nerve becomes the optic tract. From here, it becomes the geniculocalcarine tract, or the optic radiation. It lands in your visual cortex. Okay, so the visual pathways are such. Um, Visual nerve signals leave the retinas through the optic nerve. At the optic chiasm, nasal halves of the optic nerves cross to the opposite sides. They join the fibers of the opposite temporal retinas. Optic tracks both synapses in the dorsal lateral geniculate nucleus of the thalamus, and it becomes the geniculocalcarine fibers, or the optic radiation, or the geniculocalcarine tract. It now end up in the primary visual cortex in the calcarine fissure area, which is in the medial occipital lobe. Here, this is the medial occipital lobe. Okay. You have concurrent visual pathways. Aside from once it reaches the visual cortex, some fibers would be sent to the suprachiasmatic nucleus of the hypothalamus to control circadian rhythms, which synchronize various physiological changes of the body with changes of the night and day, like cortisol more produced during daytime, daytime causing you to wake up. These are the circadian rhythms of the body. Okay, um, and then some fibers would go to the pretectal nuclei in the midbrain, which will elicit reflex movements of the eyes. Focus. It will also focus on objects of importance. So if there are several things in front of you, you would focus on one object to look at one object only that is in the pretectal nuclei in the midbrain. It also activates the pupillary light reflex, which we will discuss in more detail later. Okay. The superior colliculus, it controls rapid directional movement of the two eyes together. So if it moves to the right, to the left, they are conjugate. There's conjugate movement of the two eyes. Then some fibers will also go to the ventrolateral geniculate nucleus of the thalamus, and surrounding basal regions of the brain, which will control some of the body's behavioral functions. The dorsal lateral geniculate nucleus, okay, here, dorsal lateral geniculate nucleus relays visual information from the optic tract to the visual cortex. Through the optic radiation and or through the geniculocalcarine tract, which we saw a while ago, it gives accurate, exact point-to-point transmission. Signals from the two eyes are kept apart. It is composed of 6 nuclear layers. Okay. Layers 2, 3, and 5 receive signals from the lateral half of the ipsilateral retina. Okay. And then layers 1, 4, and 6 receive signals from the medial half of the retina of the opposite eye. Also, the dorsal lateral geniculate nucleus gates the transmission of signals to the visual cortex so that after it leaves the lateral geniculate nucleus and goes to the visual cortex, some signals would go back, okay, to the nucleus. And so it receives gating controls. Okay, this one, corticofugal fibers returning in a backward direction from the primary visual cortex back to the lateral geniculate nucleus, which is inhibitory. So it gates or it analyzes the information that you want to concentrate on or to just um, not give significance to. Okay. It also receives receives signals from reticular areas of mesencephalon. Both of these are inhibitory. So when it's stimulated, they can turn off transmission through selected portions, gating circuits which highlight visual information that is allowed to pass. In the dorsal lateral geniculate nucleus, layers 1 and 2 are called magnocellular layers, which contain large neurons. They receive their input from large retinal ganglion cells and provide rapidly conducting pathway to the visual cortex. It is colorblind. It only transmits black and white information, but and the point-to-point transmission is poor. So this layers 1 and 2. Layers 3 and 6 are called parvocellular layers. They contain large numbers of small to medium-sized neurons. They receive their input from the type B retinal ganglion cells, and they transmit color. They convey accurate point-to-point spatial information, space, meaning space, moderate velocity of conduction rather than at high velocity. So this is now your medial occipital cortex. This is the primary visual cortex. The macula here, look at the macula. This is the visual cortex, primary visual cortex. Look at the representation of the macula, okay, which is very big. So there is visual acuity, visual point-to-point representation in the macula, okay? And this is surrounded by your secondary visual areas. The visual cortex is located on the medial aspect of the occipital lobes. The primary visual cortex is also called visual area 1 or V1, or the striate cortex because of its striation. And then we have the secondary visual area surrounding the visual cortex here, secondary visual areas. The primary visual cortex, it lies in the calcarine fissure area. It's extending forward from the occipital pole on the medial aspect of each occipital cortex. It terminates in, it is the terminus of direct visual signals from the eyes. Signals from the macular area of the retina terminate near the occipital pole. And as I said, there's a large representation of the macula from the retinal fovea. Signal from the peripheral retina terminate at or in concentric, so half circles anterior to the pole. Excuse me. Upper portion of the retina is represented superiorly, while lower portion is represented inferiorly. Okay. Okay, so from the primary cortex, they are now sent to secondary visual areas, visual cortex, which will now interpret what was seen in the primary visual cortex. So the visual secondary areas, also called visual association areas, lie lateral, anterior, superior, and inferior to the primary visual cortex. These are areas for analysis of visual meanings. Brodmann's area 18, where virtually all signals from the primary visual cortex pass next. It's also called visual area 2 or V2, V3, V4, and so forth, up to more than a dozen areas where various aspects of the visual image are progressively dissected and analyzed. Okay. It is also in the secondary visual area where analysis of third-dimensional position, 3D, gross form, and motion of objects are analyzed. Okay, it analyzes the 3D positions in space. It analyzes the gross physical form of the visual scene. It analyzes motion in the scene. The signals transmitted in this position form motion pathway. They are mainly from the large M optic nerve fibers of the retinal M ganglion cells, transmitting rapid signals but depicting only black and white with no color. Analysis of visual detail and color. Analysis of visual detail, dissect out color, concerned with such visual feats as recognizing letters, reading, determining the texture of surfaces, determining the detailed colors of objects, deciphering what the object is and what is. It's like when you look at a phone, okay? You know it's a phone. So the secondary association areas will be able to tell you that it's that the visual cortex sees it as a phone. The association areas will now give more detailed information like the three-dimensional figure on space and what it does. And you can say it, what this is. The visual cortex, it's just there. It's a phone. But after you analyze it, you can already say that it's a phone. Okay, so like when you look at a ball pen, you know it's a ball. Some disease we're in, you know it's a ball pen, but you cannot say it's a ball pen. Okay? The cortex just sees it as a ball pen, but you cannot say it's a ball pen. Okay, you know the meaning, so you decipher what the object is and what it means. That is done by the secondary association areas. Okay, the layers of the primary visual cortex are such. You have the retinal M ganglion cells going to you have the retinal ganglion cells which are magnocellular cells. Okay, they land on layer 4 C alpha. Okay, while the very accurate, this one is for fast transmission, black and white, while the retinal P ganglion cells or the LGN parvocellular cells, they land on the C, 4C, and 4A there. It is very accurate, interprets it as very accurately, and it has color. the interpretation sees color. Okay. Okay, so the geniculocalcarine fibers terminate mainly in layer 4. Rapidly conducted signals from the M retinal ganglion cells terminate in the 4C alpha, relayed vertically both toward the cortex. Visual signals from the medium-sized optic nerve fibers derived from the P ganglion cells in the retina, here, P ganglion cells, terminate in layer 4A and CB, C beta. They transmit point-to-point type of vision as well as color. So this is black and white, this is color. Retinal color, retinal M, retinal P color, retinal M, black and white. Okay, cortical interpretation. So this is the retinal image, it's a cross. When this image reaches the cortex, okay, it is only registered as contrast between this white here and the red. So this is what you see in the cortex. Then this will now send signals to the association areas, and you will now see that it is red inside there. So that is what the secondary association areas do. They will now interpret it as it is seen by the retina. So there is contrast, okay? Analysis of contrast is done in your, first in the visual cortex here. There's contrast between the borders and the background, okay? There, analysis of contrast. Spatial pattern of the most excited neurons in the visual cortex. So this, the spatial pattern of most excited neurons in the cortex would be the areas in the borders, okay? The maximum excitation occur along the sharp borders of the visual pattern. Here, excitation is here, there's contrast there. Visual signal in the primary cortex is concerned mainly with contrast, okay, contrast, than with non-contrasting areas. So this is not registered. The red part here is not registered in the primary visual cortex. Okay. Orientation of lines and borders. These are the simple cells detect the existence of lines and borders, detects direction of orientation of each line or border, vertical or horizontal, or lies at some degree of inclination. For each such orientation of a line, specific neuronal lines are stimulated. A line oriented in a different direction excites a different set of cells, found mainly in the layer 4 of the primary cortex, okay? So for example here, this line excites one set of neurons. This line excites another set of neurons. This line and another set of neurons at a different place. So you have spatial differentiation and contrast. The complex cells detect line orientation displaced laterally or vertically in the visual field. As the visual signal progresses further and further away from the layer 4, some neurons respond to lines that are oriented in the same direction but are not position specific. So in the detection of line of specific lengths, angles, or other shapes, some neurons in the outer layers of the primary visual columns, as well as neurons in some secondary visual areas, are stimulated. So some neurons are stimulated by lines, some are stimulated by borders of specific lengths, some by specific angulated shapes, and some by images that have other characteristics, okay? So the redness of the cross, which we showed a while ago, are detected by the secondary visual, uh, by the secondary visual areas. Okay, so the redness. Remember the redness of the cross. Detection of colors. Detection of color is also by means of color contrast. A red area is often contrasted against a green area, a blue area against a red area, a green area against a yellow area. All these colors can also be contrasted against the white area. Mechanism of color contrast analysis depends on the fact that contrasting colors called opponent colors excite specific neuronal cells. Okay, so the blue cones excite blue neuronal cells. Okay, the red cones excite certain neuronal cells. Okay, more complex contrasts are detected by complex and hypercomplex cells. Okay. The perimetry chart, this is actually to see where your blind spot is, okay? So in the field of vision is the field of vision is the visual area seen by an eye at a given instant. So you have the nasal field of vision in the middle and the temporal lateral side field of vision to diagnose blindness in specific portions of the retina. So this one, you put one chart in the one charts the field of vision for each eye by perimetry. The subject looks with one eye toward a central spot, okay, directly in front of the eye, while the other eye is closed. A small dot of light there is moved back and forth, and you are asked, okay, you are asked when the spot of light or object can and cannot be seen while the light is moving in front of the eye. Okay. So the part where you cannot see the spot, the light or the thing that is being spotted is called the blind spot because it corresponds to the area where the optic nerve exits the eyeball. And here it lacks the rods and cones in the retina over the optic disc. What are the abnormalities in the fields of vision? Okay, if you destroy the entire optic nerve, okay, so if this is optic nerve, there would be blindness of this one eye. Okay. Of the destruction of the optic chiasm here prevents crossing of the nasal fibers. Okay. We have what you call bitemporal hemianopsia. Study the figure and tell me what, and then diagnose what bitemporal hemianopsia is with the destruction of the optic chiasm. Also study what homonymous hemianopsia is, as this is the interruption of an optic tract, denervating the corresponding half of each retina on the same side of the lesion. So there is neither eye can see objects to the opposite side of the head. There, optic tract. There is destruction here, optic tract. Okay, so again, study this, analyze what vision is eliminated. Okay, there is also, read on this, is scotomata and retinitis pigmentosa. Read on these two conditions. The eye movements are controlled by the recti muscles, the lateral medial, superior, inferior recti muscles, and the superior oblique and inferior oblique. The medial rectus, superior, inferior rectus, And the inferior oblique are all controlled by cranial nerve 3. The superior oblique is controlled by cranial nerve 4, while the lateral rectus is controlled by cranial nerve 6. Okay? They would be passed through your medial longitudinal fasciculus, okay, here. They would go into your superior colliculus, and this is the one in charge of your conjugate movement. Okay, when the one eye moves there, to the right, both eyes move there, left, right, left, up, down. Okay, so there's conjugate movement. The two eyes move together in the same direction. This is a function of the superior colliculus. Um, Fixing the eyes on a discrete portion of the fixation movements. Here you try to fix the eyes on a discrete portion of the field of vision. This is controlled by two neuronal mechanisms. You have the voluntary fixation mechanism. So it allows a person to move the eyes voluntarily to find the object on which or she wants to fix the vision. Break, a little break. Another mechanism is the involuntary fixation mechanism. It holds the eyes firmly on the object once it has been found. This fixation mechanism causes the eyes to lock on the object of attention once it is found and is controlled by the secondary visual areas in the occipital cortex. Okay. But then, once you fix your eyes on a certain object, your eyes elicit certain movements. Okay. What movements are this? Okay. Um, there is continuous but almost imperceptible movement, a tremor rate of 30 to 80 cycles per second. Here, and there's tremor, okay? Caused by successive contractions of the motor units in the ocular muscles. There is also a slow drift of the eyeballs in one direction or another, small drifting, okay? Sudden flicking movements that are controlled by the involuntary fixation mechanism. So there is movement, okay, there's movement, but it is put back into the fovea by the voluntary movement to fixate it. Okay, so it goes back to the fovea for fixation. So even though you're looking at a certain object, there's active movement in your eyelid, in the eyeball, but this is fixed by the voluntary movement to fixation of the eye. This is done by the superior colliculi. This is controlled by the superior colliculi. And this results from a negative feedback mechanism that it prevents that object of attention from leaving the foveal portion of the retina. So even if it keeps moving, it goes back to the fovea. Okay, there are gross movements of the eyes. You have saccadic movement of the eyes, a mechanism of successive fixation points where the visual scene is moving continually before the eyes. It's like riding in a car. So there is continuous movement of the eye. So your eyes doesn't get fixed in one area unless you want to fix it in one area. You want to look at that area, at that object for a prolonged time. Okay. Otherwise, you go with a ride. There's saccadic movement of the eyes as you travel. Okay. There is also saccadic movements during reading. A person makes several saccadic movements of the eye for each line while he reads. You also observe saccadic movements in observing a painting, like movement of the eye upward, sideways, downward, and ungulated directions as you look at a painting, a work of art. Okay. There is also fixation on moving objects, or what we call pursuit movement. Eyes remain fixed on a moving object and it moves with the movement of the object. It jumps by means of saccades in approximately the same wavelength pattern of movement as that of the object. So if it's jumping, you move with the jumping movement. Okay. Well, how is fusion of the visual images from the two eyes done? Visual images in the two eyes normally fuse with each other on corresponding points on the two retinas. Okay. These are transmitted to different neuronal layers of the lateral geniculate body. The signals are relayed to parallel neurons in the visual cortex. Interactions occur between these cortical neurons to cause interference excitation in specific neurons when the two visual images are not in register or not precisely fused. Excitation provides a signal that is transmitted to the oculomotor apparatus to cause convergence or divergence or rotation of the eye so that fusion can be reestablished. So there is movement of the eye. So there is fusion of the object in the visual cortex. The corresponding points of the two retinas are then in register. Excitation of the specific interference neurons in the visual cortex disappears thereafter once you have an in register of objects in the visual cortex. Okay. What is strabismus? It's also called squint, cross eyes, or lack of fusion of the two eyes. So there's no fusion of vision of the object in the cortex. Sometimes this is corrected by first putting a patch, okay? Putting a patch over this while she's still a child. Or maybe there's a weakness of the lateral rectus here, or very strong medial rectus muscle. So surgical correction, okay? So this squint, strabismus, or lack of fusion could be sign that there is an unequal pool of muscles on the eye. Stereopsis or depth perception is seeing an object, seeing objects at different distances. Okay, the neural mechanism of judging distances of visual objects is called stereopsis. Okay, because the two eyes are more than 2 inches apart here, the images on the two retinas are not exactly the same. The right eye sees a little more of the right side of the object. The left eye sees a little more of the left side. Like I cannot see what's here. Okay, I cannot see what's here with my right eye. Okay, the closer the object, the closer the object, the greater the disparity. Pathways from the retinas to the visual cortex stray 1 to 2 degrees on each side of the central pathway. Some optic pathways from the two eyes are exactly in registers for objects 2 meters away. Another set of pathways is in register for objects 25 meters away. The distance is determined by which set of pathways are excited by non-register or register. So if you are seeing two objects, one is 2 meters away, the other one is 25 meters away, the 25-meter object registers in this area of the brain, of the retina, okay, and the brain, and this one registers in a certain area of the retina and in the brain. Okay, so here. So the distance is determined of the pathways are excited or non-registered. Okay, like this one there. This, halimbawa, ito yung nasa 25 meters away, it falls here. This is at 2 meters away, it falls there. So it is perceived as nearer, farther. Okay. And You can only judge the distances up to 200 feet or 60 meters. Beyond that, you can no longer see if it's farther or nearer your eyes. The autonomic control, okay. Parasympathetic is the cranial nerve 3 to 5. Sympathetic here from the thoracic segments. Okay. Okay. Parasympathetic control fibers arise in the Edinger-Westphal nucleus. Edinger-Westphal nucleus there. Then pass through the cranial nerve 3 to the ciliary ganglion, which lies immediately behind the eye. There, ciliary ganglion. And then it synapses with postganglionic parasynaptic neurons, which in turn send fibers through ciliary nerves into the eyeball. Okay. These nerves will now act on the ciliary muscle that controls focusing of the eye lens for accommodation. Remember the discussion a while ago regarding the ciliary muscle contracting, releasing their hold on the lens, making the lens more rounder, less flat. And then it also acts on the sphincter of the iris that constrict the pupil, which is in charge of the pupillary reflex. Okay, for example, you put a light, you can see this doctors testing the pupillary reflex by, like, there's a light there, point it to the pupils, and what is the reaction? The pupils constrict. So light is suddenly placed in front of the eye. This sends messages through the cranial nerve second, cranial nerve 2. It goes to pretectal region. It stimulates the cranial nerve 3 from Edinger-Westphal nucleus there, nerve 3, and it constricts the pupils. Okay? There, that's a pupillary reflex. The sympathetic innervation, it originates in the intermediolateral horn cells of the first thoracic segment. Okay, and the sympathetic fibers enter the sympathetic chain, pass upward to the superior cervical ganglion. It synapses with the postganglionic neurons. It reaches the eyes. It innervates the radial fibers of the iris, which open the pupils. So it causes sympathetic innervation, causes dilatation of the pupils. It also innervates several extraocular muscles of the eye. We have seen earlier that the parasympathetic is the one in charge of accommodation. How again? It contracts the eye ciliary muscle, relaxes the hold on the lens, making the lens more convex, increasing the refractive power of the lens. Okay? Relaxation causes decreased power. Lens changes its strength in the proper direction to achieve a new state of focus within a fraction of a second. Okay? So let's now have an example, okay? So here, I'm looking at an object, hairbrush. No, this one, a yellow. Yellow lighter, red. This red tie. Okay, so anyway, this lighter, this one, it's less, okay. So as it goes nearer my eye, any object as it goes nearer, my lenses would try to accommodate or become more convex so that I can see it clearly. But I have to use glasses because, as I said, I'm starting to be presbyotic, okay? And then, okay, so here again, so let's look at this. The red cones stimulate, the red cones are stimulated when I look at this. So I see red. Red cones, green cones, and blue cones are stimulated when together at a same magnitude when we see white color. Okay, so that's it. And then, okay, so this one, okay, is seen in the retina as upside down. It registers in the retina as upside down. But the moment it goes to the brain, this is interpreted by the brain as upright, same as this one. So in the retina, it's upside down. Okay. In the brain, it's upright. There. Okay. What else? What other examples can we have? Okay. So this one, okay, the borders are interpreted, the lines are interpreted by the cortex, but the association, visual cortex, primary visual cortex, but the this one, the area there is interpreted by the association area, secondary association areas. Okay, orientation, uh, first visual cortex, and then orientation, 3D, all are interpreted by the secondary visual areas. Okay, it is a lighter. I know it is a lighter, and I can say it is a lighter. These are all a function of visual cortex association, secondary association areas, and the motor areas, which will now say this is a lighter. Okay, so I hope I was able to lecture lecture well, and for you to understand the physiology of vision. So if you have any questions, I don't know how you will ask me, and I can answer you. Maybe we can have a Zoom meeting so that we could have an answer. We could have a question and answer portion. Okay. So um, happy reading, happy listening, reading, and listening to my video.